New e-guide, from lead to investigational new drug, successful preclinical studies with Gattefossé excipients. Drug development is a long and risky venture, which is mainly attributed to high costs and late project failure. The purpose of our new guideline is to provide researchers with effective formulation strategies and non-clinical safety data of our excipients. This allows the characterization and optimization of the pharmacological and safety profile of a compound for oral drug delivery. The guide helps you rapidly select the most appropriate excipients to early assess the developability of your compound. And when time is short or formulation knowledge is missing, we suggest ready-to-use platform formulations for rapid in vitro or in vivo screening. To assure correct study results, we also inform you on the safety and tolerability of our excipients and adequate dosing volumes for frequently used test species. Finally, we summarize all relevant excipient information in summary tables. And this is how the guideline starts. We list all our excipients potentially enhancing the systemic exposure of your compound. It describes the excipient type and the suggested mechanism of action after oral dosing. Overall, transcutol is the only non-lipid in our range. It is a solvent and improves solubility without showing any biopharmaceutical activity. All other Gattefossé excipients are lipids. They are digested and not only improve but also maintain the solubility of your compound in the post-digestive phases. Examples are given in proof-of-concept studies from the literature. They show that Gelucir, Labrazole and Labrafil can be very efficient on their own when orally administered to different test species. This efficiency is related to their self-emulsification and digestion into mixed micelles in the body fluid, and to the improved absorptive flux of the dissolved compounds. When combined in lipid-based formulations, the effects of the single excipients can be improved. Since lipid formulations can be long to develop, we provide ready-to-use platform formulations from the literature that were proven already efficient in vivo. We also recreated these formulations in our laboratory and collected additional data for stability, dilution capacity, and dosing recommendations. You can use these platforms to readily screen the solubility or in vivo performance of your compound to readily assess its developability. The guideline also gives an overview on the safety and tolerability of our excipients after oral administration, in a summary table at a glance and in detail afterwards. When available, intravenous exposure levels are also provided. This allows you with the same excipient obtain the absolute bioavailability of your compound. It is very important to handle lipid excipients correctly. They are esters and therefore prone to oxidation. Correct handling ensures reliable results. The guide contains both an at-a-glance overview and more detailed information for the specific case of Gelucir 4414, a semi-solid excipient. A useful dosing table is provided to avoid failure of repeated dose studies. For example, Labrazole, being a very efficient solubilizer and absorption enhancer, has very bad taste and induces emesis in dogs when administered alone. The guideline provides useful tips to overcome these challenges. A summary table in the end of the guide highlights all relevant excipient properties, enabling your rapid retrieval when needed. We recommend you start using our e-guide by reviewing the summary page of key excipient properties, dosing, handling, and possible effects of following oral administration. You can easily zoom into this page, which is designed to help you find the most relevant information to your study and get the most out of the e-guide. We hope that our guide will become a practical tool in your everyday work, and we would be delighted to get your ideas on further improvements. To obtain a copy, please contact your local Gattefossé office.